Good afternoon, everyone. I'm Dr. Reshma, Assistant Professor. Today's topic for small group discussion is malaria and cysticercosis. Coming to today's topic, we'll deal under following subheadings. Contents are malaria, introduction, causative organism, life cycle, morphology, laboratory diagnosis, cysticercosis, causes, etiopathogenesis. Coming to introduction of malaria. Malaria, as we know that malaria is a major public health problem in tropical and subtropical developing countries of the world. It is endemic in African and sub-Saharan region of the world. 300 to 500 million cases have been reported yearly and 1 to 2 million deaths are due to malaria. India reports around 1 million malaria cases annually. Coming to causative organism, malaria is caused by bite of female Anopheles mosquito. There are 4 species which causes malaria: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malaria, and mixed infection. Mixed is caused by Plasmodium vivax and Plasmodium falciparum. Vivax accounts for 70% of the infections, and Plasmodium malaria accounts for less than 1% of the cases. Coming to life cycle of the malaria. The life cycle of malaria involves 2 species. A definitive host is a mosquito and an intermediate host is a human. Asexual phase of the cycle takes place in human and the sexual phase cycle takes place in a mosquito. Exogenous sexual phase in mosquito is called sporogamy, during which parasites multiply, and the endogenous sexual phase in human is called physiogony. I repeat, life cycle of malaria takes place in two species: human vertebrates, that is an intermediate host. A sexual phase of the cycle takes place, and a sexual phase takes place in mosquito, which is a definitive host. Exogenous sexual phase in mosquito is called sporogamy, while endogenous sexual phase in human is called physiogony. Liver cells and RBCs are involved. The process of reproduction in protozoa, where production of daughter cells occur by binary fission, begins when infected female Anopheles mosquito bites a person and trophozoites are injected into circulation. There are 4 stages in human cycle: pre-erythrocytic physiogony, erythrocytic physiogony, gametogony, exoerythrocytic physiogony. Now we'll deal with all this one by one. That is 4 stages in human cycle: pre-erythrocytic physiogony, erythrocytic physiogony, gametogony, exoerythrocytic physiogony. Transmission. The transmission of malaria occurs by bite of female Anopheles mosquito. When a mosquito bites, sporozoites are liberated in the bloodstream, and these circulates for about 30 minutes and invade the liver cells. In liver, these sporozoites undergo development in liver and multiply by nuclear binary fission. Second, pre-erythrocytic phase. It is also known as tissue hepatic phase. This takes place in hepatocytes. Sporozoites mature and sessions which rupture and release the merozoites. What happens in liver? Once the mosquito bites, these sporozoites will circulate and liberates a sessions. Sporozoites multiply by nuclear divisions and liberate 20,000 to 50,000 merozoites. And the incubation period of each species is different. For falciparum vivax, it is 8 days. For Plasmodium falciparum, it is 6 days. For Plasmodium malaria, it is 13 to 16 days. For Plasmodium ovale, it is 9 days. The hypnozoites get stored in a liver cells in vivax and oval, which are responsible for relapse of the disease. Merozoites liberates in a bloodstream. Coming to erythrocytic phase, a sexual phase, physiogony. Merozoites invades the red cells, and here they grow and mature into trophozoites, which appear as a ring form. These trophozoites develop into seizons. The infected red cells then rupture and release a numerous merozoites from the seizons to infect the other red cells. The merozoites release leads to fever, chills, rigors, and other symptoms of the malarial infection. The merozoites liberates in a bloodstream, whereas P. vivax affect the young RBCs and malaria affects the old RBCs. Falciparum affects all the RBCs. These merozoites are pear-shaped, 1 to 5 micron in length, and these have got a receptor which are present on RBCs, glycoprotein. The liberated merozoites are in three forms: trophozoites, ring form, seizons. Coming to sexual phase, which takes place in a mosquito. Some of the merozoites which differentiates into male and female gametocytes form a plasmodium infective to the mosquito. These are taken up by the mosquito during another blood meal. They fuse to form a ookinete in a gut lumen of the mosquito. The ookinete invade the stomach wall of the oocyst. This in turn develops and releases sporozoites, which migrates into salivary gland of the mosquito, and this mosquito then goes and infects the another human host. To summarize, this is the photograph showing the malarial life cycle where both human, an intermediate host, and a definitive host mosquito. It is a cycle which takes place both in human and mosquito. Malarial parasite life cycle involves two hosts. During a blood meal, malaria infected female Anopheles mosquito inoculates sporozoites into the human host. These sporozoites, what happens, infects the liver cells, and these sporozoites mature into seizons, which rupture and release a merozoites. In vivax and oval, there is a dormant stage of hypnozoites, which can persist in liver if untreated and causes a relapse by invading bloodstream a weeks or even a years later. After this initial replication in liver, exoerythrocytic physiogony, the parasite undergo a sexual multiplication of erythrocyte, that is known as erythrocytic physiogony. Merozoites infected red cells, the ring stage of trophozoites, mature into seizons, which rupture and release the merozoites. Some of these parasites differentiates into sexual erythrocytic stage, that is gametocytes. Blood stage parasites are responsible for clinical manifestations of the disease. The gametocytes, male microgametocyte and a female macrogametocytes, are ingested by female Anopheles mosquito during a blood meal. The parasite multiplication in mosquito is known as sporogonic cycle. While in mosquito stomach, the microgamete penetrate the macrogamete, generating zygote. These zygote in turn become a motile and elongated ookinetes, which invade a midgut wall of the mosquito, where they develop into oocyst. The oocyst grow, rupture and release the sporozoites, which make their way into mosquito salivary gland. Inoculation of sporozoites takes place, and this cycle repeats. Coming to clinical features of malaria. Stage 1. There are three stages: cold stage, hot stage, sweating stage. Cold stage, which lasts for about 15 minutes to 1 hour, patients will have a severe chills caused due to rupture of host red cells which escape into blood. presents with nausea, vomiting, and headache. Stage 2, it is a hot stage. Patient will present with a high grade fever which lasts for several hours after starts of invading new red cells. Stage 3, sweating stage. Patient presents with sweating, concludes the episodes of cycles are frequently asynchronous paroxysm which occur every 48 to 72 hours. In malaria, pyrexia may last for 8 hours or more. Temperature may exceed for 41 degrees Celsius. I repeat, clinical presentation takes place in three stages: cold stage, hot stage, sweating stage. In cold stage, patient will present with chills, which may last for as short as 15 minutes or longer duration, it may extend up to 1 hour. It is caused due to rupture of host red cell which escape into blood. And with this, patient may present with nausea, vomiting, headache. And second stage, it is hot stage. Fever may reach up to 40 degree and may last for several hours, which starts invading newer red cells. Third is sweating stage. Patient starts with sweating, concludes the episodes of cycles, and asynchronous paroxysm appears. Coming to incubation period. Incubation period of malaria is 9 to 14 days. It is nothing but the appearance of the first symptoms after the bite of female Anopheles mosquito. Clinical features. Early symptoms. Patient may present with fever, chills, sweats, headache, nausea, vomiting, body ache, generalized malaise. Usually appears 10 to 15 days after person is infected. There are characteristic fever pattern in the malaria. P. vivax causes benign territarian malaria. It is benign as it is rarely fatal. Territarian as a fever is on every third day, that is after 48 hours. Relapse may occur because of dormant hypnozoites which reside inside the liver. P. oval infection has periodicity and relapse similar to P. vivax, but it is milder and can be cured. Falciparum malaria causes quaternion malaria. It has 72-hour cycle, no exoerythrocytic stage, but relapse may occur. P. falciparum causes malignant territarian malaria. Malignant as it is severe form of malaria. Territarian as a fever occurs every third day. Infected red RBCs form a cluster called rosettes. Such rosettes may block a capillaries of vital organ causing renal failure and encephalopathy. These are the following fever pattern in a malaria. P. vivax causes benign territarian malaria, which means that fever occurs every third day, 48 hours, and there may be relapse because of dormant hypnozoid stage which resides in the liver. P. oval infection has a periodicity and relapse similar to P. vivax, but it is milder and can be cured. P. malaria causes quaternion malaria. It has 72-hour cycle fever. Once in every 72 hours, fever appears. There is no exoerythrocytic stage, but relapse may occur. In falciparum malaria may cause malignant territarian malaria. Malignant as it is severe form of malaria. Uh, Territarian as a fever occurs every third day. Infected red cells form a cluster called rosettes. Such rosettes may block the capillaries of vital organ causing renal failure and encephalopathy. Splenomegaly. With the repeated paroxysm, there is a significant enlargement of spleen, secondary to activation and hyperplasia of mononuclear phagocytic system. Initial changes is congestion leading to soft enlargement. Later, it becomes dark due to accumulated malarial pigment. Diffuse cellular hyperplasia, dilated sinusoids, and accumulation of macrophage attenuates the enlargement of spleen, which becomes hard due to fibrosis. Splenomegaly, it is seen in 30 to 40% of the cases. Coming to complication which occurs in severe falciform malaria, which have manifestations of cerebral malaria, patient may present with coma, hypoglycemia, renal impairment, severe anemia, hemoglobinuria, jaundice, pulmonary edema, acidosis, followed by congestive heart failure and hypotensive shock, out of which cerebral malaria is very fatal disease. Coming to morphological features which are caused in various organs due to malaria. Parasitization and destruction of erythrocytes are responsible for major pathological changes as under. Malarial pigment liberated by destroyed RBCs accumulates in the phagocyte cells of the reticuloendothelial system, resulting in enlargement of spleen and liver, which leads to hepatosplenomegaly. What happens is malarial pigments which are liberated due to destroyed RBC. These starts getting accumulated in phagocytic cells of reticuloendothelial cells, and these accumulated cells results in enlargement of spleen and liver, leading to hepatosplenomegaly. In falciparum malaria, because of excessive absorption of hemoglobin by the renal tubules, which producing a black water fever. This is also known as hemoglobinuric nephrosis. Coming to autopsy finding, at autopsy cerebral malaria, it is characterized by congestion and petechiae on white matter of the brain. The parasites erythrocytes in falciform malaria are sticky and get attached to endothelial cells, resulting in obstruction of capillaries of deep organs such as brain, leading to hypoxia. What happens? These parasized erythrocytes in a falciform malaria become sticky, get attached to endothelial cells of the vessel wall. When these parasites attach to endothelial cells, and these results in obstruction of capillaries. These obstruction of capillaries of the deep organs such as brain. When obstruction leads, it leads to hypoxia and death. If a patient is present on CT, you can see microhemorrhage and microinfarcts may be seen. Coming to laboratory diagnosis of malaria. Laboratory diagnosis of malaria, complete blood picture shows anemia. Anemia is nothing but reduction in hemoglobin. Following are the reasons for the cause of anemia in malaria. It is results of destruction of parasites, red cells, suppression of erythropoiesis, immune hemolysis. Patient may present with microcytic hypochromic or microcytic normochromic anemia, pronounced in falciparum infections. Leukocyte count. Leukocyte count with a recurring febrile paroxysm leukopenia may occur. Leukopenia is nothing but reduction in total WBC count, which may be as less as 3000 to 5000 cells. Neutropenia, decrease in neutrophil count less than 50%. Monocytosis, increase in monocytes 10 to 20%. Monocytes and neutrophil contain ingested hematin pigment. In WBC, total WBC count is reduced, neutrophils are reduced, and monocytosis will be seen. Platelets, there is a reduction in platelet count, which is known as thrombocytopenia. Causes for thrombocytopenia in malaria is sequestration in spleen, immune mediated lysis, dysbiotic process in marrow with a diminished platelet production. Coming to the pictures of malaria, these are the photomicrographs showing the peripheral blood smear with the falciform vivax species of malaria. Here you can see the ring stage of falciform vivax, and here amoeboid form of vivax. This is the other photomicrograph showing a ring stage of falciparum, and this is gametocyte of the falciparum infection. Pause. This is the gametocyte form of the falciparum infection. Coming to blood film examination, there are two types of blood film examination in malaria: thick blood film and the thin blood film. The importance of thick blood film is it will helps to tell whether the parasite is present or no. It is used for detection of malaria in larger volume of blood is examined, allowing the detection even at low levels of parasitemia. Thin blood films, it gives which type of species is present. It gives more information about the parasite morphology and therefore is used to identify particular infecting species of the plasmodium species. Blood films remain a gold standard for diagnosis of malaria. It is highly sensitive, possible to detect malarial parasite at low densities, help to quantify the parasite load, distinguish the different species of malarial parasite at their different stages. Threshold for detection in thin film is 100 parasite per microliter. In thick film, it is 50 to 20 parasite per microliter. Coming to different modalities of diagnosis, there are several other modalities which have undergo development in diagnosis of malaria. Rapid diagnostic test, detection of antigen, immunochromatographic dipstick method, parasite histidine rich plasma and parasite LDH based assay, PCR based technique, fluorescent techniques, Kawamoto technique, quantitative Buffy code technique, serological tests like ELISA, immunofluorescent assay, radioimmunoassay, gel diffusion, and the culture. Coming to morphological feature recognition of malarial parasites. This is a pictorial representation, and this red color represents the RBCs and the blue is the cytoplasm, and here it is the chromatin dot of the malarial pigment. This is the schematic representation of erythrocytic stage of malaria. This is the RBC ring form, blue cytoplasm, trophozoites, sejons, and gametocyte. This is the photomicrographic picture of malarial parasite of erythrocytic stage. Here it is ring form. Here you can see a ring with a chromatin dot, and these are the sejons, approximately around 20 to 24 trophozoites, and these are the gametocytes and these are the trophozoites. This is the photomicrograph of Plasmodium species, Plasmodium falciform species of malaria. These are the various forms which you can see in peripheral smear. These are the double chromatin dot applique form, multiple infections in a same RBCs, and these are the ring form, and here you can see gametocytes, mature and immature form, and these are the trophozoites and these are sejons. 8 to 24 merozoites are seen in the peripheral smear. These are the various forms of Plasmodium falciform which you see in peripheral smear. Coming to vivax species, in vivax you'll see Schaffner's dot, ring form, trophozoites, deformed erythrocyte, amoeboid form, merozoites, 12 to 24 merozoites, sejons, gametocytes. These are round to oval. These are the various forms of Plasmodium vivax. Coming to Plasmodium ovale, infected erythrocytes moderately enlarged, fimbriated oval Schaffner's dot, ring forms. Trophozoites are compact, sejons, 16 to 14 merozoites, gametocytes. These are the Plasmodium malaria. Trophozoite is compact. Trophozoite typical form, band form, sejons 16 to 12 merozoites and gametocytes. Following features are seen in peripheral smear. Coming to other modalities, diagnosis by fluorescent microscopy. Fluorescent microscopy, it is modification of light microscopy. Fluorescent dye detects a RNA, DNA that is contained in parasites. The nucleic material is not normal in mature RBCs. Kawamoto technique is used. Stain thin with the acridine orange. Requires a special equipment. Fluorescent microscopy is needed. Stain itself is cheap and sensitivity is around 90% by the fluorescent microscopy. Coming to quantitative Buffy coat. Fluorescent microscopy after centrifugation, acridine orange coated capillary is filled with 50 to 100 microliter of blood. Parasite concentrate below the granulocyte layer in the tube. May be slightly more sensitive than a light microscopy, but few reports have shown 55 to 84%. Quantitative Buffy coat is useful for screening large number of samples. It is quick, saves a time, requires centrifuge and special stain. Three major disadvantage of quantitative Buffy coat is species identification and quantification is difficult, high cost of capillaries and equipment, can't store a capillaries for later reference. Coming to serology, malarial serology, antibody detection, immunological assay to detect a host response. Antibodies to a sexual parasite appear some days after invasion of RBC and may persist for the month. It is not useful for routine diagnosis of active infection as they cannot distinguish current and past infection. This antibody detection has valuable epidemiological tool for retrospective survey in endemic areas, useful for identifying infective donor in transfusion transmitted malaria, investigating congenital malaria, especially if mother smear is negative, diagnosing or ruling out tropical splenomegaly syndrome, retrospective confirmation of empirically treated immunes. These are the serological applications of antibody detection in malaria. Coming to antigen detection, immunological assay to detect a specific antigen. There are commercially available kits as immunochromatographic rapid diagnostic test, falciform histidine rich protein, and parasite LDH. Rapid diagnostic tests are based on detection of circulating parasite antigen. There are two, that is rapid histidine rich protein and parasite LDH. Coming to polymerase chain reaction in malaria diagnosis. It detects a very low level of parasites in blood, that is less than 5 parasites per microliter of blood with very high sensitivity and specificity. Threshold for parasite detection is zero point 0.1 parasite per microliter if a whole blood in a tube, or 2 parasite per microliter if using filter paper. Definitive species specific diagnosis is needed. It can identify mutations and try to correlate to drug resistance. Coming to real-time PCR, new technique based fluorescence, promising because it has potential to quantify parasitemia, may detect multiply wavelength in a same tube, identify the multiple species in one run, saves the hands on time, needs a further research and validation for malaria. Coming to useful of PCR in malaria. Diagnosis of malaria, following response to treatment, epidemiological survey, screening of blood donor, research tool in malaria control program to carry out quality control checks on microscopic diagnosis. Coming to drawback, high cost, need a special equipment, and it is a lengthy procedure, time consuming. This is all about the malaria. Now we have seen about introduction, causative organism, life cycle of malaria, clinical features and lab diagnosis. Coming to cysticercosis. Cysticercosis is an infection caused by larval stage of Taenia solium, the pork tapeworm. Adult tapeworm resides in human intestine. Eggs are passed in a human feces, which are ingested by pig and or they infect the vegetables. These eggs then develop into larval stage in a host, spread by blood to any sites in a body or form a cystic larvae termed as cysticercosis cellosis. Human being may acquire an infection by larval stage by eating undercooked pork or by ingestion of uncooked contaminated vegetables, sometimes by auto-infection. This is the picture showing a life cycle of cysticercosis. People eat, when we eat a raw or undercooked pork containing a cyst tapeworm larva, cysticerci, in intestine, cysticerci mature into adult, which attach themselves to the wall of intestine. Adult worm produces segments that bears a egg called proglottids, which may release egg or detach from the tapeworm. These egg or detached proglottids are passed into stools. Pig or people are infected by the ingesting egg or a egg bearing proglottid, and these egg develop into spears which invade the walls of intestine, and these spears travel to the muscle or other organs and develop into cyst. This is the life cycle of cysticercosis. Coming to morphological feature of cysticercosis. This cysticercosis may be single or there may be multiple cysticerci in different tissue of the body. The cyst may occur virtually anywhere in the body or accordingly produces symptoms. Most common sites are brain, skeletal muscle, skin. Cysticercosis consists of round to oval white cyst about 1 centimeter in diameter, contain milky fluid and invaginated scolex with a bifringent hooklets. The cysticercosis may remain viable for long time and incite no inflammation, but when an embryo dies, it produces a granulomatous reaction with eosinophils. Later, lesions become scarred and calcified. This is the photograph showing numerous cysticerci at the base of the brain. This is the photomicrograph of skeletal muscle, cysticercosis in skeletal muscle. The worm is seen in the cyst, while the cyst wall shows palisade layer of histiocytes, and these are the cuticles, body of parasite, and this is the coelomic cavity. Thank you.